Hello, good afternoon. It's really a pleasure to be here once again in our webinar. And this week we are starting the series of microbiota. And we have a pleasure to have with us today Professor Selosse. Thank you so much for your time. Thank you so much for staying with us this afternoon.
You're more than welcome. It's my pleasure.
So, Professor Selosse, I met not you really is the first time we are meeting, but I met your research in a book that have been given to me, and I really like the way you write because for me you are able to translate the science and poetry, and I really love the way that you put to the science in an easy way and a very beautiful way. And then I know that you wrote another book that I didn't read yet that is about microbiota. Can we start to talk a little bit about your background and about this microbiota book that you have?
Well, when I was young, I was having interest in fungi, mushroom picking. Like 8, I was 8. Okay, and when I went into my studies, based on that, I went into more mushroom, mycelium. I realized that the daily life of mushroom was microbiological or microbial because they are the hyphae, the mycelium are something very teeny in soil, in plant. And then I woke up as a microbiologist. At some point, I went to be a microbiologist. I was first teaching at University of Montpellier in southern France, and then I recently, 2013, joined the Museum of Natural History in Paris, partly because one of my goals was not only to go on teaching and doing research, but also to do more outreach, because I think in the current days, science is accumulating more data than society does use. I mean by that we have to spread more the news, and also the society has to listen more to some things, some discoveries that are very important tools for the future. And microbes are very important potential tools for the future of plant health, animal health, or also human health.
Yes, what I really like when I start to read your book, it's about because I'm very fan of Dr. Lynn Murray. I love her. I think that she was like the beginning of all this changing when you talk about microbiology and evolution. So I would like you to, because normally the public that stay with us, they are nutritionists, they work with animals, and we talk a lot about microbiota lately and try to understand how the microbiota could be used in agriculture and animal productions. But I would like your concept, like use a little bit of the symbiosis concept of Dr. Lin, about what Holobiont is and how is the symbiosis concept in terms of sustainability.
So I think there are 3 concepts to introduce. The first one is that in nature, when you have 2 species, they can interact in different ways. One can exploit the other. This is parasitism in the broad sense, including predation, when one is killing the other to eat it, which is an extreme case of parasitism, of course. So this is plus, minus. One benefits, one pays. You have minus, minus. It's competition. Competition is harming everyone. And we have to think of this because very often in economy, we see that competition or concurrence is doing a great job. It's simply killing resources. For biologists, not for economists. I let you with this idea. Minus, minus, competition. And the third one, I only discuss the most relevant one in ecology, is the plus, plus. This is mutualism, where the being together of those 2 species is positive, but they are not necessarily always together. Think of a bee pollinating a flower. It's a plus-plus interaction. The bee is eating, the flower is pollinated, but they are not living together. They just transiently interact. If you have mutualism and permanent interaction, like in a lichen is a kind of teeny crusty organisms made of an alga, living with a mycelium of a fungus. or when you have the sea anemone with the clownfish, then their interaction is positive. The fungus is feeding the alga is feeding the fungus. The anemone fish is protecting the fish, and the fish is protecting the anemone, and the anemone is giving some waste to the fish. So it's a positive interaction, but they live together. It's a subset of mutualism that we used to call symbiosis, from the Greek, living together, symbiosis. Okay, so mutualism with this specific case of symbiosis where the mutualists are together. Don't need to join to be together and have the benefit of the interaction. Finally, another idea was recently emerging. I think it's a very weak concept, but it's fashion. It's the holobiont. The holobiont is the idea that when you look at me, or the plant behind me, or sorry, Elizabeth, but looking at you or the plant beyond you, you think you have one organism. It's totally false. It's totally false. What you have is a big one, which is a place where many others are living. In me, for example, there are as many microbial cells as human cells. If you take 1 gram of leaf of the plant behind us, you will find 100 millions of microbial cells, mostly bacteria, a bit of fungi, a bit of yeasts. So it means that those big organisms are also having symbionts. We will discuss together what they do exactly and why they are beneficial, but to say that it's not an organism, it's something which is interacting with a lot of things, and we will discuss how necessary this interaction is. To account for this, we say, okay, this is a microbial community in a plant, or this is a microbial community in Elizabeth. And so this community with the big host, which at first glance we see, but which is hiding a diversity of interactions, this community with a big one is called a holobiont. From the Greek holo, all the bionts, all the living things together. But you know what? It's symbiosis. Just sometime when you rediscover something, you like having a new name. But symbiosis was invented in the end of the 19th century, and we just discussed symbiosis here.
Oh, perfect. This is amazing. So let's talk a little bit about to people understand better this concept. For example, for animal production, we have like the nutritionist, and they normally do the best. And now we talk about microbiota in these animals. So they always think that we can have like a modulation of the microbiota, especially in the gut microbiota, with the nutrition, like the way that they put the vitamins, minerals, and all this kind of stuffs, or essential oils and plants, whatever. But for example, I'd like to discuss also the concept that comes before the nutrition, because this feed comes from plants. So can you explain to us like the microbiota growing in plants, in soil, and how this all is connected up to the end in animal and human microbiota?
Yeah, it's the big picture, but it's a bit difficult to do. But well, let's face it. The health and nutrition, the immune system, and the nutrition and the digestive systems of plants and animals is always helped with microbes. You take a plant, the roots of the plant are not always able to extract nutrients from soil. They need fungi which associate with their roots. There's association of a root with a fungus. You would call it a root because with your eyes it looks like a root, but under the microscope there are hyphae, microscopical hyphae of fungi. So the fungus is in the soil and in the root. The plant is having root and leaves, and this root with fungi is called a mycorrhiza, which in Greek means, sorry, there's a lot of Greek. It means root fungus. It's a mix of the two, and most roots, or most of what you call roots, are mycorrhiza, and they are very necessary for extracting nutrients from the soil. By the way, Elizabeth, just one thing, truffles, for example, are an example of fungi that are living on the roots of trees. That's why you are harvesting the truffles under the trees. Okay, I insist on this symbiosis because this is the symbiosis on which my teams at the museum in Paris, at the Kunming University in China, and the last one in Poland, in Gdańsk, my research teams in the 3 countries are working on this association. But also, the plant is green. And you know what is green in plant, which is not green in me? Plants have small microbes in the cell belonging to the cyanobacteria. You know, they are in every cell. So at the first glance, people thought that those were cell part of the cell. But indeed, what is green? The chloroplast, as we call them, which has the chlorophyll and is performing the photosynthesis. Yes, it's a part of the plant, but it was a free-living bacterium that once entered the ancestor of plants. So photosynthesis is a bacterial thing. So you see that for food, fungi and bacteria are providing the food to this plant. Okay, now let's take you and me. Our digestion, it can be also animals of any kind. Our digestion is helped by microbes. And not only that, but also the vitamins we gain are due to bacteria. For example, when you grow mice without any bacterium, we call them axenic mice, you see that they need 30% more food to have the same growth increase. They digest less well. And they regularly need pills of vitamins, which is bizarre, because they have a deficiency in vitamins because they have no bacteria. There is a final touch that you and me and rabbits and mice, we respire. In our cells, there is a small sub-thing of the cell which is performing the respiration. This is called a mitochondrion. And mitochondria are also bacteria that are in our cells since generations, since the common ancestor of rabbits, mice, humans, and even plants, by the way, because they also have that. They respire in the night, you know. And so there's the respiration is a kind of, it's due to a bacterium employee of the cell. So you see, exactly like the chloroplast of the plant, this is inherited. This is within the cell. But some other symbionts, like the mycorrhizal fungi or the bacteria of the gut, we gain them during our development. We gain them from the environment because a seed, an embryo in a seed, or a newborn are sterile, more or less sterile. So we have to gain it from the environment. So you see, this is nutrition. And now, to make it brief, this is also protection. Because all those organisms, they have an evolutionary interest for their host to be fit, because they receive some food from the host. The fungi on the roots, they receive sugar from the plant, those mycorrhizal fungi. The microbes in my gut, they receive the food I'm taking. So they have an interest to the health of their host. And over time, every of those microbes that was able to help the health got selected because it was making healthier hosts. So it was a kind of self-interest to do that, evolutionary speaking. Let me give you 2 examples. For example, the mycorrhizal fungi, they protect the roots. They allow the roots to be healthier because they are there, and I will tell you what they do exactly later. The same is true for our gut bacteria. You may know, and I know because I have a newborn at home now, that the first weeks are difficult with a lot of digestive problems and a lot of diarrhea. This is just because at the beginning we don't have this help of bacteria from the gut to whip out the negative, the pathogenic bacteria, and so we are more sensitive to that. How are those microbes doing? 3 things. First, they occupy the field. So when a new thing is coming, it's crowded already. There's no space available when pathogens arrive. So it's competition for space and resources. But the second thing is that they are sometimes themselves making antibiotics to kill the competitors. So when a pathogen is coming, it's facing a layer of antibiotic. For example, you, Elizabeth, have a huge probability of having on your skin, on your nose, Staphylococcus aureus, which is a very pathogenic bacteria. But in the nose or on the skin, there is many other bacteria. And we know many of those bacteria which are making antibiotics active against Staphylococcus aureus. So there is competition, there is antibiosis, but the third way is the most fascinating. The third way is that they manipulate those microbes. We call them often microbiota to mean the whole diversity of the species which are there. And for a human, it's between 500 and 1,000 species on the skin and in the gut. Those microbiota, they manipulate our immune system. They make it more efficient. We know this because we can compare this axenic mice, which have no bacteria. They are grown axenically without any bacteria experimentally. And we can compare them to normal mice. And we can see that when they meet a pathogen, they are dead, even if it's not a pathogen of mouse. So the third way is that they are increasing the efficiency of the immune system. They are like a signal of, okay, you are born, you are germinating if you are a plant. Now you have to set up at highest level your immunity. Nutrition and defense for plants and animals. Really the same stuff.
We have a question here from Pietro. Pietro said he asked us, ask you, is Holobiont a fancy name for genotype and environment, plant, animals, etc. interaction?
Holobiont is a way of acknowledging that as a part of the environment, a part of the living environment is inside. And it's very important because, you know, it is inside since generation. So your body and mind, Elizabeth, they are trained, they are selected to work with it. That's why, by the way, relying on microbes to set up the efficiency of the immune system is possible because any generation was colonized. You are colonized for sure. So there is a part of the environment which is in us. And now we have some habits in animal growing, in farming, but also in growing plants, or also in terms of hygiene for humans, that is reducing this level of microbes. If you put pesticides on a plant, you will kill the bad microbes, but also the good ones, or some of the good ones. When you take antibiotics or wash too much the hands in every condition, well, nowadays there is a good reason with COVID.
Yeah.
But some of my students, you know, they touch a book and they take gel, you know, to wash their hands. And why the hell? So when we overwash, when we have too sterile food, we have a decrease of this microbial diversity. And with this decrease, the functions are not very well performed because we have much less diversity of microbes than our ancestor was selected to live with. And we know now that many illnesses from the immune system, from the metabolism like obesity, diabetes, for the immune system, it's asthma or allergies, or even some behavioral illness such as autism are partly linked to the reduction of diversity of microbiota, which in certain genotypes, in certain genetic background, is turning into illness. You know what? The reduction of biodiversity is a nightmare everywhere. But the first place where it's touching plant health and animal and human health, it's this internal microbiota, which is decreasing in diversity and so decreasing in functions. So yes, it's a part of the environment in ourselves that we cannot cope without it.
You know, I was wondering, last year I went to the Holobiont Congress here in Montreal. Yeah. And in that Congress, they present a very practical, for maybe for my public to understand better, a very practical work where they show the difference in microbiota when you have, for example, different harvest, or when you do like a plant, like you put wheat and corn and wheat, or when you put corn and let's say another crop, and this change the microbiota and change also the productions and also the quality of this plant. So in the terms of Holobiont, I can think that we need to work integrate, because up to now, maybe our nutritionists from the animal side, they don't really look that depends of this microbiota, depends of the management that the crop has. They could change so much these ingredients that they need to review what they are doing in terms of nutrition for the animal. But also maybe I go through away, but maybe also they are affecting differently the microbiota of the animal that's eating this feed. Can you discuss a little bit about this aspect?
Especially an issue, you're right. So let me first tell you that the Holobiont meeting is very, I was not at this second one, but I was one of the organizers of the first one in Paris. And we organized the next one together with the International Society for Symbiosis Congress next July in Lyon, France. So there will be a joint symbiosis meeting and Holobiont meeting to try to reconnect the two concepts. I told you it's the same indeed. So we organize that next July in Lyon. So anyone is welcome. If you type,
I will be there for sure.
yeah, okay. So we will meet there. It's also a nice city for food and wine, so we have to meet there.
Yeah, I know.
To be more serious with your question, it's a big issue. We know that the microbes are manipulating a lot of things. And we know, for example, that depending on which mycorrhizal fungus is in the root, you don't have exactly the same expression of genes in the fruits. That's demonstrated on tomatoes. You change the fungus on the root, and the genes are not expressed at the same level. Some even are not expressed, others are expressed, depending on the fungus. So there is a far-reaching consequence on who is in the microbiota, who is on board of the holobiont. So for sure, the quality of the plant food depends on the microbiological diversity in the plant. Now let's take the animal, because I think it's more the focus of the people listening to us. Let's take the animals. How can we hope that what the food they take will change their own microbiota? There are classically two ways, but the truth is that there is a third one which is very overlooked. First way is that when you put some food in your digestive tract, it will allow some bacteria to develop, and others will not find the substrates or the things they need. That's, by the way, why we are asked as humans, we are asked to eat more fibers. We don't digest fibers. We often say, oh yeah, fibers, it makes the digestive flow better. Not at all. It's just feeding the bacteria. Bacteria which are helping the digestion and other stuff. So you have less inflammation, you have better digestion, and by the way, one small side product is that the digestive flow is better. So this is called prebiotics. That's the idea, prebiotic, that you can give something that will help some bacteria, and ideally the best ones. Okay. And it's fiber for us. Or mother milk, by the way. Because in mother milk, there are many things that are not digestible for the babies, but which enhance the bifidobacterium and lactobacillus, which are good for health. Okay. Second way is to take bacteria which will settle in the digestive tract. Let me tell you that the digestive tract is already crowded. So it's not very sure that they will manage. There is this idea that the success of settlement is about 1 chance per million of billion of chance. So you see, the probability is not very high. But we have some treatments that we call probiotics, where we try to take some bacteria with the hope they will settle. Honestly, when you eat vegetables, I have strong doubts that the bacteria that are living on vegetables, they can find a suitable place in your digestive tract. It's much more anoxic. It's not the same food
environment.
But there is the third very overlooked story. And I don't know why it's so overlooked, because it's logical and there are evidence. The evidence is called yogurt. I think yogurt is something clear to everyone. When you eat yogurt, in yogurt there are a lot of bacteria, Streptococcus and Lactobacillus, okay? When you eat yogurt or fermented milk, it does change a lot your own microbiota. But not because those bacteria settle. They don't settle there, you know, they're living on cheese. It's not the same environment. No, they don't settle there, but they change while they go through the digestive tract. They change your microbiota. And in the case of yogurt, they're changing in lowering bacteria which are pro-inflammatory, so which produce some products that are making immune reactions and create a deleterious system with a lot of free radicals and the beginning of inflammation, a low level of inflammation, but it's never good, neither in a pig, nor a rabbit, nor myself. And they foster other ones, especially the ones which are producing butyrate. And butyrate producing bacteria in the digestive tract of most animals, they are very good because this butyrate is feeding the cells of the intestine as a substrate, but also regulating a lot the immune system. So they really enhance the health and both the reactivity, but also the lower basal level of functioning of the immune system. So it's not prebiotic. You are not giving food to bacteria. It's not probiotic. You are not giving bacteria that will settle there. We know they don't settle. So I call it very often in my teaching pseudobiotic. It's just a disturbance in a good way of the internal microbiota. This we know for human. You have my best recipes here, fibers and yogurt or cheese. We know it for human. We are far from having a good view for animals because our knowledge of the physiology of animals is a bit one step behind. And this knowledge, it's the knowledge of the effectors. And so I think it's really something, a continent to conquest now. If you are feeding rabbits or chickens.
Swine, ruminants.
Yeah, so for ruminants, we know they depend a lot on those microbes, of course.
Yes.
Whole digestion is there. So we still don't have a good view of probiotics, prebiotics, and pseudobiotic effects. But let me tell you something.
So,
yeah,
yes.
Oh, sorry.
I just ask, for example, I have a question here from my friend Claudio Cabral. He asked me, I put his questions in a different way. What you believe that could be more important to modulate the microbiota? You were saying, like, I put the microorganisms that is in the yogurt, they could disturbing, but they are not really live there. You know, so what do you think? Do you think that it's better to manipulate the microbiota in the good way or bad way, adding microorganisms on it, like probiotic, or adding ingredients, nutrients like essential oils or organic acid or fibers, like you're saying, or maybe put different kinds of vitamins, minerals, tannins, or whatever? What do you think that is really affecting more the microbiota in animals or in plants? What affects more when you think about the ecosystem and everything that happens in there?
I would say that all have the same potential to be as efficient, okay? But some are reliable, some are not. For Claudio, to be clear with what I mean, he could survive with a pouch of glucose and amino acids going into his blood, okay? He will survive. That's exactly what you do when you give fertilizers to a plant or every kind of requirement to an animal, like nutritional requirement to an animal. You substitute to the evolutionary and ecological logic that is normally giving that, you substitute by bringing yourself everything, which results in two things. First, costs, because you have to buy all those ingredients, like fertilizers for the plant or very complex nutrient for the animals. So costs. And second, we know what we can bring what we know is required, but what about what we don't know is required? We have to acknowledge that we don't know everything that is needed by a plant or by animals. Recently, a friend of mine was publishing a paper, I can't remember the molecule, but he discovered a molecule that is clearly in Europe leaking from the modern food because we didn't know it, so we don't add it and we don't have standards for that. I think it's a kind of cofactor which we don't do ourselves and we need it in the food. Sorry, his name is Danchin, so you may find the exact molecule. Cuin, I think it's cuin, but I'm not very sure. Anyway, the idea is that we will give what we know the plant or the animal need, but what we don't know, we won't give. So I don't like this idea of giving chemicals or molecules in the food or giving microorganisms. I think what you have to do, and this is something, I'm a plant biologist, this is something we are trying now to think with the mycorrhizae. Rather than putting fungi in the soil, we try to think how to manipulate the soil, to cultivate. You were mentioning that, Elizabeth, what you do the previous year, what you do in winter has an impact. So what to do to sustainably maintain the microbes that will help. And I think that we have tomorrow of thinking not to give the bacteria or the molecules always, but to have a kind of food which is self-supporting the diversity, which in turn is supporting the health. So I think that the idea of giving addition, it's not good economically, and it's not helping a lot because we don't know everything. We have rather to resume a walking of the plants and of the animals, which is by itself doing the job. That is based on a diverse microbiota. So it means we have to have less hygiene. Even if I don't ask for pathogens to come back, we need less hygiene. We need a less processed food, because when you process, you maintain some constituents, but you don't maintain the ones you don't know. So it's a plaidoyer for simpler things. It's a paradox, okay? The problem is that currently the industry is rather on the way to offer new products based on new knowledge. And I think we are going in the wall. We still do not reconnect with a complexity that we will never, never, never fully understand. So we'll never, never, never make the artificial food that is perfect. It's not possible. We will always be trying. What about resuming the normal functioning?
Do you think there is some questions here about the transfer of microorganisms, like fecal transplant, or when you take the microorganisms from one to another? I know that you are more plant, but I think in plant is the same. Do you think that this is a good strategy, or according to your answer is not, but I just driving for the questions that appears after your.
It's complex. I would say first that it's not new. Early in the 18th century, when a young calf had problem to digest after weaning, there was this use of taking from the mouth of other ruminants, of taking a bit of rumen and giving him this. And it was empirically known that he was then digesting better the herbs, and it was a transplant of gut microbiota. Shall we make transplants? Yes and no. Yes, in some case, for example, when people undergo a chemotherapy, which can be killing sometimes the microbiota, For better health after the chemotherapy, it can be good to have a fecal transfer. Okay. It can also be good in case of very heavy antibiotic, for example, before a transplant of bone marrow. So it could be good to make a transfer to restore the normal functioning. If you do that, a good thing is to do it, to think of self-transfer. That is, you make a provision before the treatment and you transfer it after. Like thinking local. And for plant mycorrhizae, it's the same. Go to the soil of the field and try to amplify the fungus there and make your own inoculum. Okay, so on the one hand, yes, it's a good idea, the transfer in some pathogenic situations. It may be that tomorrow we will have some fecal transfers also for some illness like obesity or diabetes. For now, it's a dream, but we know it has to do with the microbiota. So it can be a way of curating, but we are issuing from billions of years without any transfer. We had a biology which was doing the job. So the goal, I know it's not easy and it's not for tomorrow. The goal is to use what was ever used before we started to touch the thing in every direction. I agree that now there is some situation where we will have to do the fecal transfers. But I would like to warn you about fecal transfers. We don't know the negative effects. We have a couple of very interesting studies that show that you can have a lowering of diabetes or a lowering of Crohn's diseases, which is an autoimmune diseases. You also have very nice works on pupils, which are autists. You can, with a lasting effect for several years, you can lower their level of autism. So there are very promising ways. But those promising ways are promising in a situation where the way we are living all days is not enabling us to have an equilibrated microbiota. So it's a kind of saving the situation with the transfer, which is an idea, which is raising a lot of hopes. But the mother of all the hopes is that we will stop damaging our microbiota. And it's the same for animals. You know, there are very nice experiments that shows that the animals we are growing, the pigs, the rabbits, or the mice, we already have been communicating to them an over-hygienic situation in which they don't perform well. It's very well known for pigs that most pigs living in a very clean environment, they have a gut microbiota which is pro-inflammatory. That is more than for any pig. That is more promoting an inflammation level, which is the first step to problems, physiological problems, cancers, dysfunctioning, aggressivity. So you see, yes, transfer are a good idea, but they will not replace the logic. And let me tell you that recently 2 people died in an experimental fecal transfer in the States.
I read it, yes.
Because some bacteria were lactam, they were resistant to antibiotics and they had a septicemia. So actually, I think that 2 died and 3 were ill. And when you read the paper, 2 were ill and 1 died. But then after the paper, there was more. So it's a dangerous tool, and as any medication, it has bad effects, bad side effects, and we still didn't explore that very well. So before I can answer this question, we need more case to know in which window of opportunity it's a good idea. But nevertheless, it's only because we damaged the microbiota before.
You know, the time is passing so fast. I would like to stay longer talking to you because really it's interesting. But I'm taking like for Holobiont is that I learned today that you need to have like a balance between everything that we are doing as humanity in this planet. Like we should understand that when you change the weather, when you change the soil, when you put antibiotic, every movement we are changing the disturbing. I use your word. You say the word disturbing this Holobiont system. And so you need to be very careful about that. And I think that today I feel, I'm in R&D, so I feel that people are very pressurous, at least myself, I feel sometimes pressures to find something that could be like the the solutions to solve all these microbiota modulations, to have more productions and to have more health. But as much as a study, as much I understand that this is a concept that could be balanced between many things, because we know, for example, the environment, the behavior, the stress, the temperature, the weather, everything make difference. And when you talk to the animals, we need really to put together all this information for us to really have a good response about that. So as we are going to the end of our conversations today, I hope I can have another opportunities to stay more long with you. I'd like you to give us your final message and mention a little bit about your book that you have wrote about the microbiota, what this is about, for us to have more to look at in your
Yeah.
view of microbiota.
I'm sorry that the book is only in French. The French is never alone because we are always in company of microbes, and the subtitle tells you everything about the book. It's those microbes which are building plants, animals, and civilizations. Now, the take-home message is the following one. We've been missing the microbes and their role because they were too small. We only saw them because of illness, and we had this bad idea to think that they were only linked to illness. If you read carefully the work of Pasteur, Pasteur wrote somewhere, an animal could not survive without microbes. Pasteur had a much broader view of that. He knew that they were pathogens, but he suspected that they were important. And nowadays we realize that they are beyond every functioning of the organism. And the problem is the following one. On the short term, if you have an ill animal or an ill plant and kill everything, the plant is saved. So we have many procedures that are efficient on short term. The problem is that we lose the help of the microbes because we've been killing the good and the bad ones. And the problem is that all this over hygiene, over abuse of antibiotics, abuse of cleaning, this is killing the day after, and on the long term, you make simply the plant or the animal more susceptible to metabolic or pathogenic diseases. So we have been growing an agriculture and a farming, which is very efficient on the short term, but just by killing the long term. And by handling more the microbes, we should act now, today, by not only solving the problem of today, but not only we have to solve the problem of today, but also we have to act in a way that is solving any problem on the long term as well. And the microbiota are a door to that.
What's your final message about microbiota, Holobiont? What we should go through? Final message for all these people that work with animal production. How can we see the microbiota? Like, how can we see our job to manipulate it?
I know more the human side of the story with the probiotics, and we are working with some colleagues on regulation now, because I can see that there is a lot of products that are now available. On the one hand, in the future, those products, well, even if it's not perfect, the transfer, the probiotics, they may be a solution. But many of those products, there is no regulation, there is no rules, no labeling. So you have, if you want to use them, to be very careful, very, very careful, whether it's a serious product with a lot of research behind, or whether it's just a sell of something that may make you happy, but will not be very active. What I mean here is that a good product, there is always written somewhere where it's not good and what are the risks. If a product is good for everything, I think you can buy candles and go to the church. I think you can buy 17th century wonderful medicine that was sold by wonderful doctors. Look at the seriousness of the technical notice. It should say this is demonstrated good there and there. Don't use it there and there. We don't know, or it's even bad. Like for a medicine, because microbes, they can cure us. So exactly as medicine, they may also damage us.
Yeah. So we need to be careful about the relationship with
And
them.
full of hope, but full of hope, open to novelty. Yeah.
Thank you so much for your time. I really appreciate all your effort to be here. I know how busy you are. And thank you everybody that stay with us. And remember, we have this series for more 3 weeks. And just to finalize, I have a question here for Maria Therese. She wants to know when and will be the next Holobiont Congress. Maybe you can just
Thank you
finish
for
saying that.
advertising a lot again. July of next year, if COVID is allowing us, July 18th to 30 to 23, 18 to 23 in Lyon, France. So Lyon is a city from east of France, close to the Alps.
Very
Welcome.
good. So everybody will be there, and for sure we have a very good discussions. Thank you so much, and have a good afternoon, good night for you in Europe. Thank you so much.
Merci.
Have a nice
Thank
day.
you a lot.
Bye
Bye-bye.
bye. Merci. Merci beaucoup.